Thank you very much for inviting me to speak. I'm really excited. A little nervous after Eugene, especially given what I'm going to talk about, but we're going to go with it. So I'm really excited to share with you some is more recent, some is data that we've been holding on to for a long time, most of the reason being that it's hard to describe some of these RNA viruses and how we're capturing them. But I'm going to try to highlight some of our work. So also I don't know how to, is this how I go forward? Got it? Okay, so I'm going to go over the overview of what I'm going to speak about. So I'm going to start with a little brief introduction and then highlight some of the global data sets that I'll be sharing with you, and then talk about a little bit of the data in kind of vignettes, so really just highlighting some of the data and how we're thinking about this in terms of looking at the spatial distribution of RNA viruses, as well as putting some of this sequence data into a phylogenetic sort of approach, into thinking about them, specifically looking at population size dynamics, and also talk about event timescales, which are important for the organisms that we're going to be talking about, and how these timescales are important for links between host stress and viral infection, as well as how we can try to look at how viruses are really impacting the larger food web. I'm going to follow up with some of our culture work, where we're working on developing a new system in the lab to evaluate some of these processes that we're seeing in these large environmental data sets, and then finish by highlighting some of our next steps. So I am going to speak about one group of RNA viruses today, those that infect the marine single-celled eukaryotes called diatoms. And so the viruses of diatoms that have been isolated so far have all been is this a pointer too? Have all been single-stranded DNA and single-stranded RNA. And so these are some of the smaller viruses that we know about on this end of the spectrum in terms of genome size, and I'm going to speak just about the single-stranded RNA today. And there's only about 6 or 7 that have been characterized, some of those from the environment. And so hopefully our data will kind of push this forward into these new territories about these viruses. So one of the first questions that I always get is how many RNA viral sequences are present in seawater, and do we have enough? Are we sequencing at enough coverage so we can really start to examine these viruses? And so, go back. So one way that we're doing this is we're using a protocol that Marianne Moran published earlier, and this is incorporating spikes into our sample processing, so that we can go back and try to quantify certain gene sequences in our data sets. And I also want to point out that the data sets that I'm going to be talking about today are all from a cellular fraction. So they're all the diatoms and those viruses that are associated with them that we've passed on to a filter. We also sequence the viromes, and those are really important to continue to sequence. They're a little bit more difficult to handle, but they do give us more ideas and estimates in terms of turnover and evolution of viruses in the systems. So when we do this analysis, when we look at some of the spikes that we add into our data set, this is just one data set shown here, and we look at the conserved RDR P, the RNA-dependent RNA polymerase gene of RNA viruses, we see that, at least for this data set, we're getting 10 to the 7 RDR P sequences per liter of seawater. And it generally, between all the data sets that I'll be showing you, it's about 10 to the 6 to 10 to the 9. So we definitely are getting pretty high levels of these sequences in all of our metatranscriptomic data sets. So where do these data sets come from? I'm going to show you primarily data sets that are shown, all of the data sets actually, that are shown in red on this slide here. And this includes libraries from 302 different libraries. Of the whole data set is actually 495, but some of the new data I'm not comfortable sharing yet, but we're getting there. And we have a lot of shipboard experiments, we have bottle experiments, and we also have environmental or samples from environmental sample processors. So these are in situ sample processors, and I'll talk about this a little bit further. So really trying to examine how these viruses are interacting with their hosts and what they're doing in the environment by using a bunch of different methods to analyze them. So first, one thing that we've been working on with collaborators is also trying to first design a really robust RDR P gene tree of the reference sequences. And we're working with Miroslav Obernik on this. And so he and his team are generating some really nice phylogenetic trees that we can then use with all of our environmental data sets. And this becomes important because one thing that I mentioned is that we don't have a lot of representatives in this databases of these viruses that infect diatoms. And so one way that we kind of get around that is that we do a lot of phylogenetic-based analyses, and one of those is shown here, where we basically take this reference tree, and then we do phylogenetic placement of all of our environmental sequences on these large trees, and we can look at the diversity and what we would more likely annotate them as using this method as opposed to something like sequence similarity, because a lot of times what you'll get is these diatom viruses that are in this clade. This is the diatom clade here, and if you just do similarity, you're going to get honeybee and cricket and all kinds of other things, but really they're not. They're most likely diatom viruses based on where we're sampling them as well as where they phylogenetically been. So taking that phylogenetic data, we started thinking about, is there any kind of spatial distribution within all of these different libraries and all these different data sets that we can see, and is it within and between these environmental regimes? And so one thing that we originally started looking at was, can we capture this in the phylogenetic structure? So can we get population structure that's captured in the phylogenies that kind of gives some clue as to maybe some kind of genetic bottleneck that's occurring due to geographical patterns. And so, of course, when we started looking, these are RDR P sequences from all of the environmental RNA virus genomes that we've been able to pull out of these data sets. Obviously, it's kind of very difficult to look at. They're very diverse, and the phylogenies are not very strong yet. So we've started using a different approach, looking at mapping. And so here what we've done is we've taken all of those environmental genomes that we have assembled out of these data sets and all the reads that have mapped through those from the individual data sets, retook all those reads and put them against the different genomes, and then looked at kind of the genomic heterogeneity or homogeneity based on where the reads were mapping from. So was it the same data set where we actually sampled and assembled that genome, or was it from a different data set? And so when you take all that data together and you do something similar to what's shown here, or well, there's some colors not showing up, but that's okay, and a multidimensional scaling analysis, you take all that data, you definitely do see that certain data sets, for instance, this Gulf of Mexico data set down here, seems to like to stay within itself, right? And then we have other ones shown here that seem to be just like a mish of a bunch of different data sets. Interestingly, a lot of these are those where we have high levels of upwelling, so these bloom-bust dynamics. So one thing that I don't think I mentioned about diatoms is they're really elegant and they love to bloom, and they also love to outcompete a lot of the other algae in the system, because as soon as those nutrients and temperatures become right for them, they just immediately increase their biomass and their numbers and their transcriptional expression, which is really fun to think about, especially in terms of viral infection. So looking a little deeper at that coverage, those coverage plots, I just wanted to show what some of it looks like. There's a lot here, but this top part is a composite, so this is basically all the different genomes lined up, and then you see the coverage from the same data set in black and a different data set in red. And below is just an individual example of a genome. And so I don't want to go over all the data, but one thing that was interesting that we started seeing in these is we see these punctuated sort of regions, right, of really high coverage across the genomes from different data sets. And we're kind of starting to bin these out and to look at these different regions and to think of them as maybe conserved regions that's kind of adding to this ubiquitous nature of these sequences and of these genomes. And can we use that to anchor some of our annotation to? So can we then go back to our environmental data sets? Maybe we can even pull out new RNA viruses that we just couldn't annotate well the first round. So we're still working on this, but it is interesting that we keep seeing these little punctuated instances of mapping from all the different data sets. And so basically what we're seeing here is that there does seem to be some kind of geographic limitation on certain data sets, but that's not true for all of the different data sets that we're looking at. And so another aspect, again, is thinking about these blooms and how that might imprint itself on kind of the viral history. And so one way of thinking about that, one way we've been kind of thinking about that, is asking the question, do sites with this greater spatial heterogeneity, right, so those ones that we typically saw in the Pacific Ocean that are upwelling, do they have more genetic diversity that could be linked to the host bloom dynamics that's occurring? And so one way we've been trying to think about this is in kind of a phylogenomic framework, which is used a lot in human virus studies. And one aspect of that is looking at population size, where population size can, I guess, be imprinted on the terminal branch length. And so I've shown some of the data sets here. This is Gulf of Mexico, the Pacific Ocean upwelling, and the Monterey Bay upwelling. And so the Pacific Ocean upwelling was one that we actually targeted. So we saw on satellite data that it was upwelling. It was creating diatom blooms. We went out and we sampled it. The Monterey Bay is a different kind of upwelling and bloom situation in that we actually made it occur. So we took Monterey Bay water, we took deep water, and we added it to surface water, and then we monitored the bloom through time. But for each of these that we know we had, for each of these that we know that we had a bloom going on, you do just see by eye that you see an increase in terminal branch lengths in terms of the diatom biomass. So we have these host biomass going up, and we also have these are RDR P trees again. So we have the viral populations and their terminal branch lengths increasing. But if you extract that data, so if you pull out all the branch lengths as shown here, interestingly, the Pacific Ocean upwelling was actually quite a bit higher than the Monterey Bay, even though there's a big difference between the host biomass that's present. So our whole thinking was that as you increase biomass and as you're having these bloom dynamics, you would increase these branch lengths, but it doesn't seem to be the case. And it's, we think it might be because of diversity. There's probably other metrics that we need to pull out of the data, but we do see that, and for the Pacific Ocean upwelling, that the diversity, I keep hitting the wrong button, the diversity of the sample is higher, I guess I can't go back, is higher in that middle one as opposed to the Monterey Bay in terms of richness as well as diversity estimates. So thinking along these lines and thinking along the bloom dynamics, one of the major issues with these kinds of data is that we don't have the appropriate timescales a lot of times, right? We don't have temporal data sets, or we don't even know what the timescales are. So we have some laboratory studies, especially from Cathoceras and their viruses, that we know kind of the timing of events that happens in a laboratory setting, but nothing really from the environment. So what we have started doing is now going back to some of these same areas where we've done some of those large metatranscriptomic data sets, and we have included what's called an ESP. So this is an environmental sample processor. And so it's an in situ processor. It filters in situ, it preserves, and then you can trigger it to multiple times a day. You can trigger it based on nutrients. You can trigger it any way that you want to, that you have connected to it. And then you go out and retrieve it when you're finished. So you can get really nice fine scale time resolution of your samples. And so we've done this twice. I'm only going to show data from this first one, which is in Monterey Bay, because we have the sequence data back. This was done over a month in Monterey Bay, multiple time points per day, sometimes up to 4 time points per day, triggered at varying levels of nitrate. And then we also have a much longer deployment in the upwelling of the Southern California coast, which is a 4-month deployment, and we have a bunch of different types of sampling going on here to try and evaluate what's happening with the viruses through time and how they're interacting possibly with their hosts. So for the Monterey Bay data, here is a little bit of it shown here, some of the major players, and here's just a stylized picture of what's shown over here. So we have the diatoms in green, diatom viruses in red, and nitrate in orange. And so what we see in here is that we see, as we would expect, a diatom response to higher nitrate. Right here, you see this nice diatom peak happening. Then we get viral infection putatively, right? Because we have all this increase of specifically diatom viruses. This is used using that phylogenetic placement that we've shown before. You then see a drawdown of the diatoms and a secondary pulse of nitrate right here, right? But this time the diatoms don't seem to be able to respond as well as they did the first time. So this is really fun data for us to look at because it's really the first that we have where we get to see these viruses in a food web dynamic, right? So we're really looking at them through time and finally getting a picture of what's going on. We also get to extract some timescales out of here. And obviously we want to look at the data that we get out of here before we say this is true. Between when we see these increase in diatoms and an increase in viruses, we do see that happen on a 2-day time period. But the time between the two peaks is actually 6 days, and the time from the top of the RNA peak to where the RNA viruses for diatoms have finally diminished, that's 12 days after that. So it's much longer timescales than we actually thought previously. So it's fun to try and think about this maybe even from a modeling perspective. So during these time series, we have a couple of temporal data sets that I'm going to try and highlight some of the data from, but we see this same dynamic every time where we see an increase in diatom transcription, and then we see a following increase in the diatom viruses. And this is not true for double-stranded DNA Phycodiniviridae that also infect algae. It's actually the opposite for those guys, which is pretty interesting. And every data set we've looked at seems to have this same this same attribute to it. So thinking about this, it kind of makes you think about kind of what's going on, right? There's host stress and maybe a link to infections. So we started looking back at some of our temporal data sets. So this, again, is the Monterey Bay upwelling, where we've mimicked the upwelling, where we've added that deep nutrient-rich water to surface water, and then we've taken replicates and time points at the start of this exponential phase, so when they're responding to nutrients, as well as at the very end when they're nutrient limited. So they're stressed right at the end. And one way that we evaluate stress is down here. Again, this is another one of our reference trees. These are diatom light harvesting complexes, and then we've mapped our environmental sequences on there. We know that diatoms express this one in yellow, this light harvesting complex here in yellow, preferentially when they're stressed. And so you can see that in these data sets, or these samples that come from the green, so the nutrient limited, you see by and large, mostly it's just all green that's coming down here on this yellow one. So yes, they're stressed. So then we felt good that we could look at the viral sequence abundance in terms of this viral stress. And so what we see, of course, is that we do see the same thing where we have an increase in RNA viruses. The diatom viruses, I should say, are related to the Picornavirales. And we see them primarily, almost exclusively, in the nutrient-limited samples, as opposed to the Phycodiniviridae, which I mentioned before. These seem to be higher in the replete samples in all the data sets that we look at. So just kind of looking at this another way, this was obviously just over a 3-day period and very limited data set. Well, let's go back to that ESP where we've looked over a whole month, and let's see, do we have evidence of host stress, and do we also have evidence of these diatom viruses that seem to be responding after the diatoms themselves. And so if we look at this data, now we're looking at so this is looking at the response to nitrate, right? So viruses or diatoms love nitrate. As soon as they have enough of it, they love to bloom, and then they also become very limited very quickly. And so just for a comparison, I have the chlorophytes up here, so other types of algae here that seem to be higher expressed. So if they're on the positive end of the graph, they're basically differentially expressed at high nitrate. Diatoms actually have a slight differential expression towards the nutrient limited, which we know that they kind of do this last ditch effort of all their genes that are responding to this stress right before they kind of go kaput. But in terms of the viruses, what we see is that almost exclusively the diatom viruses, I think there was actually only one sequence that came down here that was differentially expressed in high nitrate. They are expressed, or at least found, because we don't know if they're genomes versus others, at the low nitrate. So when the host cells, their host is presumably stressed. The opposite is true for the chlorophyte viruses. So the double-stranded DNA are here in blue, and actually the chlorophyte virus is about 69% of this wedge here. So it's a pretty interesting, stark dynamic that's happening that seems to be very different among the different viral groups that are present in these samples. I wish I had time to talk about all the DNA viruses because it's quite interesting. So basically, where are we now with some of these environmental data sets? So we know that we have these varied spatial distributions of genotypes, and that it seems like there's these conserved genomic segments that might be popping up that maybe we could use in downstream analysis. We also see that blooms lead to this exponential growth that we can somewhat identify. We think we need to do some more examination of this, but identify phylogenetic signatures based on branch length, those terminal branch lengths, and that it doesn't necessarily, it's not entirely dependent on the biomass of the host or even the biomass of the virus, but it seems to be there's a diversity factor there. Bloom dynamics also lead to these issues, right, of what are the appropriate timescales, and so we're trying to use some of these really large-scale environmental data sets that we have to tease those timescales apart. Also, these environmental time series really provide a lot of information about the population and community dynamics that we haven't seen before, and it gives us a lot more data towards food web dynamics, which is something that we're very interested in our group. And also that host stress, in terms of specifically in this case diatom host stress, resulting from either nutrient or environmental limitation or even viral predation, I guess, leads to increase in RNA viral sequence abundance. And so what we wanted to do also kind of in tandem was to use environmental data along with laboratory data. So we're trying to develop a new system for diatoms and their viruses in the lab, and I'm going to talk a little bit about that guy. So what we've been doing is we've been going out along the coast of Southern California, a lot of the different areas that we've also sequenced with the ESP and other large environmental metatranscriptomic data sets, and we've been isolating diatoms and growing them in the lab, various different groups of diatoms. We have focused, and what I'm going to talk about today is just one of those. This is Cylindertheca, which is a pinnate diatom. Cylindertheca is an abundant cosmopolitan diatom. It was found pretty much all over in the Terra Oceans, not necessarily the highest, the most abundant, but it is found in a lot of different regions. It has a benthic as well as a Platonic lifestyle. It's also found in chlorophyll max, not in chlorophyll max. I mean, it's really quite a unique pinnate diatom. It also is fun because we can transform it in the lab, so we can do genetic manipulation with it in the lab. And we can also, it undergoes sex, and we can replicate this in the lab as well. So this is opening up new avenues for us to investigate sexual reproduction and viral infection as maybe a mode of kind of absorption or other aspects of viral infection that we know nothing about for diatoms. We don't know how they get in. We don't know when they get in. We just know that we see them. So we're using this diatom, and so what we've done is we use micromanipulation to pick single cells from the environment, and then we culture those single cells. We grow those in defined media, and at the same time that we're picking the samples that we ultimately culture for the Cylindertheca, we also take viral samples. So when we come back to the lab, we do several rounds of viral infection before we finally feel like we have selected viruses that are infecting Cylindertheca, and then we go through time course studies. And so one of those is shown here, where we have 5 strains of Cylindertheca, where we've added viruses. The control is in blue and the treatment in red, and we see that at 72 hours is when we add the viruses. We inoculate with viruses, and 48 hours later, similar to what we see with Cathoceras, or similar to what other labs have seen with Cathoceras, we see a sharp decline in the host. So looking at this data a little bit, you can see really nicely, actually, that what the cell looks like during this. So this is the control cells. You see them pretty happy, and the treatments are obviously not very happy at this point. They have diminished cell structure, and they're almost completely gone at this point. We have TEM of the virus shown here. It is a small viral particle, approximately 20 nanometers, similar to the other RNA viruses that have been found for Cylindertheca. And I'm just showing one here, but we have several. Now that we've isolated, they're similar. So we, of course, wanted to look at the genome of the virus as well as understand a little bit more about the host, right? These are environmental isolates, so we also need to look at the 18S, at least the 18S sequence. So for the virus, though, we've also been working at the same time in our environmental, along with our culture work, we've been seeing something that has intrigued us, and these are these high molecular weight bands in our RNA gels when we're doing our RNA transcriptomic work. And so what we've been able to do is we've been able to design methods in the lab to pull these higher bands away from the lower ones and sequence those separately. And this was basically a way we were thinking, well, how can we increase the coverage of our RNA viruses away from their host? Because the hosts do wash out the sample. You get several million, I want to say maybe in one data set you might have 100 million diatom sequences and you have 10 million viral sequences. So still a lot, but it's much more diminished compared to the host. And so greater sequence coverage can give you more leverage for certain types of analyses. So anyway, so when we did this and we mapped back to, these are the Cylindertheca viral genomes that we pulled out, some of the initial ones, and we mapped these different sequences from both the low and the high. You see that predominantly the RNA sequences are coming from this high band. So now we've gone back to our environmental samples. It seems to be working well there too, and we seem to have increased sequence coverage of these RNA viruses. So the genome is similar to those that have been described previously, shown here in terms of its genome structure as well as its length for the Cylindertheca virus, and using, again, our favorite sequence, the RDR P sequence. We've been looking at the phylogenies of these. It clades very nicely with other diatom viruses. However, there is what we think is, it seems like little congruency between the host and viral phylogeny, meaning that we have these viruses that don't seem to clade at all in any specific way, but the hosts do. And so we do have quite a diversity among the hosts that I'm not going to talk about, but it's pretty interesting that we're able to capture this. And so we think that this might suggest that there's little genetic bottlenecks, at least in this system, that would be due to seasonality or dispersal limitation. And this does make sense because, like I mentioned, Cylindertheca is very cosmopolitan. I can go out any day of the week at any time of the day, and I can isolate it pretty readily, which is great for all of the other experiments that we're going to be doing that I'm just going to highlight here. In the lab, so our future directions, where we're looking at single-cell transcriptomics of infected cells. So we're doing this both at the environmental level as well as in the laboratory. So we're going to go through and we're doing complete transcriptomes using more traditional methods that we have in-house at JCVI, flow cytometry, RNA transcriptomes, and we're also using a new method with collaborators called Dropseek. We're also evaluating, again, like I mentioned, sexual reproduction and viral infection to see what's going on there. For the single-cell transcriptomes, we've been lucky enough to be able to sequence our Cylindertheca. So we've sequenced one of our main ones that we use most frequently in our viral host interactions, and this is work done by Sean Paulson, who's here, but at University of Delaware, using PacBio. And so this is going to be the fourth, I think, Diatom genome that will be out. If it comes out like I think it is right now, it's looking pretty good. And we will, of course, it's going to be highly useful because the Cylindertheca and the others that are sequenced are not very similar. So the others that are sequenced are lab rats mostly, and they're not really relevant in terms of some of the environmental questions that we want to answer. And so we're also working with Karsten Zeigler, who is doing a modeling component of all of this data. So basically what he's going to do is kind of try to do some genome metabolic reconstruction using all of the transcriptomes that we provide, as well as look at viral evolution through the different time courses of infection, as well as subsequent infections. So our future directions in terms of our field work, we're obviously increasing the sampling that we're doing with the ESP and other types of in situ samplers. This gets us the appropriate timescales, I think, for looking at viral host dynamics from both a population level as well as a cellular level. We're also increasing our spatial distribution as well. So we're really trying to sequence right off the coast of San Diego, there is the Calif, which is an observatory that goes out. It's gone out over 70 years now. And we started taking omic samples about 4 years ago on each of their cruises through this transect. And so we now have very large data sets from right out of our back door to finally really look at what happens with these food web dynamics that I kind of mentioned. So I highlighted a little bit about Dropseek. I just wanted to show it because it's so cool and I can't believe it's actually working. But so Dropseek is a microfluidic method for isolating and sequencing single cells for transcriptomics. It's been used in human cells, but when I cold called them and said, hey, you want to do an environmental guy with me? And they kind of looked at me like, what? They did it though, and it worked, and they are really excited about the data because it's something very new and fresh for them. But so this is what happens. Basically, you take your single cells and you have these beads with barcodes attached to them. You incorporate both of those into a drop. You then lyse the cell. All those RNAs are attached to the barcodes, the beads. You can then wash the beads and sequence them, so you can do tens of thousands of cells in one run in less than a day. It's amazing. So what we have so far is some initial data where this is a single Cylindertheca within a droplet, within a bead. And so what this is our collaborators at Harvard, Laura Bertolin, primarily, and who's been doing this work. And so what she's been able to do is not only do whole cell, single cell transcriptomes, which is what I've shown here, she's also been able to isolate the nuclei of these diatoms, which is incredibly important when you think about viral infection, right? Because we're going to get nascent RNAs. So we'll be able to see almost in real time, if we do it properly, what happens as soon as viruses either infect or replicate or both. So it's been super fun to work with them, and also super fun that it's actually working. So for our first pilot experiment, we used just control cells for now. And they estimated there were 23,000 beads with sequences on them. Not all of those make it through the bioinformatic pipeline, but yes, we do have several thousand RNA transcriptomes from single cells from one culture now. And so the next step, we are sending next week our first viral infection to send that to them, and then we'll be able to look at viral infection through these different experiments. In addition, as part of this project, we are just starting to work with a group at UCSD where we're developing microchemostats to also look at single cells and to monitor them through time while they're infected with these RNA viruses. I think I'm running out of time, so I'll just go forward, because basically we're using all of these single cell techniques to really get at kind of those fine scale population genetics and dynamics that we haven't really been able to do yet. So there's a lot of people to thank, obviously, on this project. The Moore Foundation is funding all of our future work on the single cell transcriptomes, and we're very thankful for that. And so with that, I will say thank you.